So we are on Chapter 5, Microbial Metabolism. And so metabolism. Metabolism is simply the buildup and breakdown of nutrients within a cell. And all of these different chemical reactions will provide energy and create the substances that sustain life. So this is just a basic explanation of what metabolism is. So although microbial metabolism can cause disease and food spoilage, such as fermentation, most of these pathways are beneficial. Most microbes, at the end of the day, take-home message, most are not pathogenic. The kind of joke I always tell my students in the lab is, nothing's really going to hurt you here, just don't lick anything, because, that's kind of gross. But generally speaking, most of the microbes that exist on us, around us, are beneficial. They're working with our bodies, with the environment. There are the few that are pathogenic, and those we do focus on because we are pretty disease-centric. Excuse me. So the different metabolic reactions, we have catabolism. Catabolism is breaking down. So when we break down complex molecules, this will provide energy and the building blocks for anabolism. Let me just let somebody in. Okay. So catabolism will break down something complicated so that we can use it to build up. And anabolism will use energy, and it will have those building blocks to build complex molecules that are required for life. It's an endergonic reaction. So metabolic pathways, these are big sequences. They are actually really, really huge. And there's whole sciences built around these pathways. There's sequences of enzymatically catalyzed chemical reactions in a cell. And these metabolic pathways are determined by enzymes. So enzymes they're encoded by genes, so you can turn genes off, turn them on. And these enzymes in every step of that pathway are going to regulate whether that pathway continues to move forward or if it stops. Say the gene is blocked, the enzymes no longer synthesize, your pathway will stop. So here we have an animation.
As organisms live and grow, they're constantly in the process of making and breaking bonds in molecules. Metabolism is the sum of all the chemical reactions that take place in an organism. Metabolism includes catabolism and anabolism. Catabolism includes the processes that break down complex molecules into simpler molecules while harvesting their energy and storing it, usually in the form of ATP. Anabolism includes the processes that build more complex molecules from simpler molecules. The energy acquired through catabolic processes is used to drive anabolic processes. Neither catabolism nor anabolism is completely efficient, so at each step, some of the available energy is lost into the environment as heat.
And we're not going to go into all of that loss of energy, heat, that sort of thing. That's for higher classes. But just very basically, this metabolitic process of breaking down and building up, it's going to be done in different ways for every microorganism. Excuse me. So enzymes. We're going to talk a little bit about enzymes and the way that they work because they are so key to metabolism. So before we get into enzymes, we start with the collision theory. You may or may not have heard of this in your chemistry class yet. The collision theory states that chemical reactions occur when atoms, ions, and molecules collide. So when you have something just in a beaker, you put your chemicals in, they're going to randomly collide and create a reaction. Now, when you think of that in a biological system, it's not so random. We can't have it just be so random. And so activation energy is the collision energy required for a chemical reaction to occur. That means how much energy is it going to take to get the two molecules that I need to come together, to get them together. And the activation energy without enzymes is very high. It's really a lot of work to get these things where they need to be. The reaction rate, whoops, there's somebody else. Let me let them in. Okay, so we're on the reaction rate. The reaction rate is the frequency of collisions containing enough energy to bring about a reaction. Without any help, the activation energy, when you're just talking about collision theory, is very high, and the reaction rate is very low. Oops. So without any help, you've got these molecules just floating around. It's a lot of energy to bring them together, and the rate at which they come together is going to be really slow, very slow reaction rate. So when you add an enzyme or increase temperature, pressure, concentration, you're going to lower that activation energy. It's not going to require so much. And the reaction rate will go faster. You'll have more collisions. So enzymes are really important. If you think of it this way, if you were to eat a piece of pizza, just a bite, one bite, it would take approximately 100 years for you to digest it without enzymes, because that's how much activation energy and how slow of a reaction rate you're talking about without enzymes. So let's look at these enzymes.
Imagine a chemical reaction with the general form AB yields A plus B. In order for this reaction to occur, the reactant molecule, AB, must acquire enough energy to disrupt its stable electron configuration so that its electrons can rearrange to form A and B. This energy barrier is called the activation energy of the reaction.
So it is, as the video says,
in living cells, enzymes serve as biological catalysts, speeding up reactions. Enzymes accomplish this task by lowering the activation energy of the reaction, making it easier for reactants to acquire the activation energy required to produce products.
I think he's done for sure now. So as I said, the activation energy is a barrier. It's very high. And then when you have the enzyme, it's low. So enzymes and their chemical reactions. The substrate is what that A and B were in that little video that he showed. The substrate is what is going to be turned into a product. So substrates contact the enzyme's active site to form an enzyme-substrate complex. Substrate is transformed and rearranged into products, which are released from the enzyme. The enzyme is unchanged at the end of the reaction and can react with other substrates. So I like to go back to my pizza analogy. I always use it when I'm talking about enzymes. So students who would be like in the math lab would be the enzymes, and the substrate would be pieces of pizza. I bring in a bunch of boxes of pizza. Everybody grabs one as they're eating them. They're digesting the pizza, but the person themselves, they're not changed. Okay, maybe they're full now, but they're at the end of the day, they're unchanged and they've made a product. So here are the steps.
Although every enzyme performs a different reaction on different substrates, they all go through the same general steps in accomplishing their reaction. These general steps include substrate binding, formation of the enzyme-substrate complex, product formation and dissociation, and enzyme recovery. Like all catalysts, enzymes are unchanged by the overall reaction. Now let's look at each step in more detail. The first step in any enzymatic reaction is the binding of the substrate by the enzyme. The enzyme includes an active site that can accommodate the size, shape, and electron configuration of its substrate or substrates. According to the induced fit model of enzyme-substrate interaction, a perfect fit does not form until the enzyme and substrate bind to form an enzyme-substrate complex. After the substrate is bound in the active site, the enzyme-substrate complex is formed. The shape of the enzyme may change to allow for an efficient chemical reaction, and amino acids may accept or donate protons or electrons to facilitate the chemical reaction. Once the enzyme-substrate complex is formed, and any reactive groups have moved to the right position, the reaction proceeds and products are formed. The products dissociate rapidly, leaving an empty active site. After the products leave the active site, the enzyme returns to its original configuration. Any groups that donated or accepted protons or electrons are also returned to their original state. Like all catalysts, the enzyme is unchanged by the overall reaction.
Okay, so that's a little more detail really than you guys are going to need, but enzymes have specificity for particular substrates. So let's go back again to my pizza analogy. Some people prefer a vegetarian pizza. They're not going to go over and start eating the meat lovers pizza, right? So it's the same thing with enzymes. They're very specific to what they're going to start to modify into product. And that makes sense. You don't want enzymes just chewing everything up. So most of them are very specific. There's some different ones, but we'll go into that. Turnover number is the number of substrate molecules an enzyme converts to product per second. That just means how fast is it going? How fast is it going from substrate to product, substrate to product? And there's some numbers that they're throwing in here, but you don't really need to worry about that because this is not an enzymatic class. Enzymology is a whole field, so it can get pretty intense. But really just knowing that specificity of each enzyme occurs, so they do have specificity for their substrate, and that turnover number means how fast. So naming of enzymes almost always ends in ACE. So you could be an enzyme if you just put ACE at the end of your name. And it's grouped based on the reaction they catalyze. So here are some basic groups of enzymes. So really a specific enzyme, an oxoreductase, it would have something in front of it like a hexose oxoreductase, which means it's very specific to an oxidation reduction reaction of hexose. So these are just families of enzymes, and you'd see this word after a more specific word. A transferase will transfer functional groups. We see lots of these transferases in our citric acid cycle. The hydrolase, that's hydrolysis, which means breaking using water. Lyase will remove atoms without hydrolysis. So that's a specific type of breaking. An isomerase is going to rearrange atoms around. And a ligase will join molecules, and that one's going to use energy. So all of these are groups of enzymes, and there's a lot more than just these, but these are the main ones we're going to see when we're looking at our metabolic reactions. And you'll see a word in front of each one of them to give it the specificity. What is it actually reducing, transferring, rearranging, hydrolyzing? And so when you start to see these enzyme names in glycolysis and the citric acid cycle, you can refer back to this slide and say, oh, okay, I know what that reaction is doing. Maybe you don't know specifically because, this is an introductory class, but you've got an idea of what's going on. Enzyme components. So when an enzyme is just alone, it's not going to be doing anything. It's just kind of hanging out. So it's inactive, and that's known as an apoenzyme. Once you start adding these other things into it, you've got a cofactor, which is a non-protein component. Not all enzymes require a cofactor, but many do. Or a coenzyme, which is an organic cofactor. So some examples of cofactors would be NAD, NADP. We're going to look at those in a little bit. They're on the slides, so don't worry. And then coenzymes would be acetyl-CoA. And we see acetyl-CoA come into play between glycolysis and the citric acid cycle. A holoenzyme is going to be the apoenzyme plus its cofactor. And once that's all bound together, it's ready to bind substrate and carry forward with a reaction. So here you have these cofactors that I was talking about. NAD is nicotinamide adenine dinucleotide. NADP is nicotinamide adenine dinucleotide phosphate. We also have FAD, flavin adenine dinucleotide. And then we can have a coenzyme, acetyl-CoA, or coenzyme A. Now what we're going to do with these, we're going to see these come back up, NAD, NADP, FAD. Their main job is to carry electrons and to have protons that they are going to donate into forward reactions. I've got something in my throat. That proton is an H. That's what we interchangeably will say, a hydrogen or proton. You're just going to hear that word back and forth. And that's because a hydrogen is just the one, right? One electron, one. So we're going to see these as donors later on and carriers. That's why they're so important. And coenzyme A is a recyclable coenzyme that activates substrates to move forward into reactions. Okay, so before we get into those reactions, we're going to talk about a few of these factors that influence enzyme activity. These can include temperature, pH, substrate concentration, or inhibitors. So when we look at temperature, this curve that you're looking at, we've got an ideal temperature where enzyme activity is the best at the peak of that curve, and that's around body temperature, usually is when we have that. We have specific enzymes that we use in biotechnology that we have isolated from microbes that we found in hot pools. So TAC is one of them. And it's this little polymerase enzyme that we got from a microbe that lives in a hot pool in Yellowstone Park. And its activity level is at a pretty high temperature. It actually has to be activated at 80 to 90 degrees Celsius. And so we know that it's not going to start working until we heat it up. And so this is just one of the ways we can control things in a lab. But generally speaking, in metabolics, body temperature is pretty good for a lot of these. The same is for pH. It's going to want usually a neutral pH. Of course, exceptions to that rule, and we have a whole microbiome in our gut. Our own enzymes all work at a much lower pH of around 2.5 because that's the environment that they live in. But most of these, we've got body temperature, regular pH. Now, if you get too far off of this scale, the enzymes are going to dissociate. So let's look at that, and then we'll talk about substrate concentration. So high temperature and extreme pH will denature proteins. So when you're looking at that figure on the left-hand side, you see an active functional protein. It's got those beautiful little curls. Those are alpha helixes. And then you've got some nice beta sheets in there. Well, if you get too high of a temperature or too high of a pH or too low, you're going to denature the protein, and then it's just a long string and it can't do anything. So moving on to substrate concentration. Now, substrate concentration will influence the rate of enzyme activity initially, but at one point, it's going to stop. So notice how this curve looks very different than the other curves. It has a part where it just stops. It stops, it doesn't go down, it doesn't go up. It's just reached its maximum velocity. That's as fast as the enzyme can convert substrate to product. Going back to that pizza analogy, I brought all those pizzas into the math lab. Every student very happily chose the kind of pizza they like, and they're eating. I go and I get 10 more pizzas, 10 more boxes of substrate and bring them into the room. They're not going to disappear any faster because everybody is chewing as fast as they can. We're at our maximum rate. Inhibitors. We have natural product inhibitors out in the environment that we're always looking for. They're very good to find drugs so that we can regulate metabolic activities. Generally speaking, we're trying to regulate the metabolic activities of a microbe to cure disease. So the different types of inhibitors that we can find are competitive inhibitors. And these will fill the active site of an enzyme and compete with the substrate. So in this direct competition, the substrate can't bind. I'm just dying here. And it bounces off. So the competitive inhibitor will just bind right into that active site. More often than not, we don't really want competitive inhibitors as drugs because they usually permanently bind to the enzyme. Now, if you want to kill a microbe, you definitely want to do that. But if you are trying to modify something in a living system and you don't want to kill it, you might not want to use a competitive inhibitor. So let's take a look at this short video.
A competitive inhibitor is a substance that is structurally similar to the enzyme substrate, but which does not react to form products. For example, sulfanilamide is a competitive inhibitor for the enzyme whose normal substrate is para-aminobenzoic acid, PABA. Note the similarity in molecular structure of PABA and sulfanilamide. In competitive inhibition, competitive inhibitors compete with the substrate molecules for the binding site, slowing catalysis. However, if the concentration of substrate is much higher than the concentration of competitive inhibitor, the substrate will outcompete the inhibitor for the enzyme active site, and catalysis will continue.
Okay, so those are just a couple of examples of inhibitors. And here's the figure that shows them. I mean, we're talking about the sulfa drugs right here. So these are competitive inhibitors that are going to inhibit metabolic processes of microbes. There are other types of inhibitors which are called non-competitive inhibitors. And they're going to interact with another part of the enzyme. This is called an allosteric site. Allostery just means binding anywhere on an enzyme that will change its shape. So it's kind of, I don't know, think of it as like tickling somebody and they change their shape. That's what allostery is. It's going to come in and it's going to change the whole shape of the enzyme. So it's going to bind to another part of the enzyme rather than the active site. And this is a process called allosteric inhibition. This indirectly changes the shape of the active site and it renders the enzyme non-functional. Very often, non-competitive inhibitors can be reversible. Sometimes they are irreversible. Now, again, we're talking about if you want to kill something, you want to kill a microbe, you probably want to look for the irreversible type of drug, of inhibitor. But if you don't necessarily want to kill it, you want to keep look at something in a living system, then you want that to be reversible. So here's our non-competitive.
Non-competitive inhibitors bind to a location on the enzyme other than the active site. For example, the HIV medication nevirapine is a non-competitive inhibitor of HIV reverse transcriptase. Nevirapine does not bind to the reverse transcriptase active site. The binding of the inhibitor causes the enzyme to change shape, so the active site is no longer able to perform the reaction efficiently. Since the non-competitive inhibitor binds in a different binding site than the substrate, increasing the concentration of substrate has no effect on non-competitive inhibition.
Okay, so that is inhibition. Feedback inhibition is the most common type of inhibition that we have in a naturally occurring system. And this is where we have end products of a reaction coming back to allosterically inhibit enzymes from earlier in the pathway. This is very often reversible, and it's a way to keep metabolics under control. You don't want a metabolic system to just continue at a very high rate. That uses a lot of energy, so you do need to control it. And feedback inhibition is the most common way to control metabolic pathways. All right, we are going to move on because we are only on slide 24 of 90. So I do encourage you that if you have questions and we don't really have time to get to them in lecture, please email me. I'm happy to respond. Mondays and Wednesdays, like today, I'll be running off to sample, but I'll be home in the evening to answer questions. Okay, so let's move on here. Ribozymes. So RNA can have function, which is very different from DNA. DNA is basically just a storage device. RNA can have function, and the RNA that functions as catalysts by binding to substrates and acting upon them are known as ribozymes. They're not used up in a reaction, and they're very frequently used in cells to cut and splice RNA. They're also used in protein synthesis in ribosomes. So ribozymes are a special subset of enzymes. All right, finally, we're getting into the energy production. Now that we've set you up to know all about enzymes and inhibition, we're going to get into the actual energy production itself. Oxidation reduction reactions. So this, if you have taken chemistry already, if not, that's fine, but this you'll see again. Oxidation is the removal of electrons, and reduction is a gain of electrons. A redox reaction is an oxidation reaction paired with a reduction reaction. These are often reversible. You should have an arrow going both ways in this, really. And these oxidation reduction reactions, we're going to let this guy talk to you for a second, but these oxidation reduction reactions are very important to move forward our metabolic pathways to produce energy.
Many metabolic reactions involve the transfer of electrons from one molecule to another. This transfer of electrons involves two coupled reactions, oxidation and reduction. These are often called redox reactions. Cells use redox reactions to transfer electrons from one molecule to another within or between metabolic pathways, such as the Krebs cycle and electron transport chain. In an oxidation reaction, a molecule called the electron donor gives up one or more electrons to another molecule called the electron acceptor. In the oxidation reaction, the donor molecule becomes oxidized. A reduction reaction occurs when a molecule receives electrons from another molecule. The electron receptor therefore becomes reduced. The term reduced refers to the receptor's electrical charge. Because it's gained a negatively charged electron, the overall charge on this molecule becomes more negative and is said to be reduced. You can now see that the oxidation reaction must be coupled with a reduction reaction. In other words, if a molecule is to donate an electron and become oxidized, then another molecule must accept that electron and become reduced.
Okay, so we're pairing molecules together. This is essentially the backbone of making a pathway. And in biological systems, we find that electrons and protons are removed at the same time. And that's equivalent to a hydrogen atom. So we're always going to see, whenever we find an electron donor, we're going to also see the removal of that hydrogen. And this is very important when we get into the electron transport chain. We're going to see our cofactors pop up again, that NAD and FAD. They're going to have H's on them from the citric acid cycle. And when they move into the electron transport chain, we're going to see the removal of that H and the donation of an electron. So this is very important when we move into that reaction. Biological oxidations are often dehydrogenations. So this is the backbone of how we're going to create energy. So let's look at these exact systems, and you're going to see these little reactions pop up in the big metabolic pathways to generate ATP. And ATP is generated by the phosphorylation of ADP with the input of energy. ADP, that D stands for di. So it's adenosine diphosphate, and that means 2. So when we want to have high energy phosphate bonds, we usually want 3 ATP. Most reactions are going to cleave off that final phosphate high energy bond and use it if they need energy. So we want to create a triphosphate, a 3 phosphates, in order to create energy. The different ways that we can phosphorylate to make that ATP. So ATP is generated when that high energy phosphate is added to ADP. So substrate level phosphorylation is just simply putting that phosphate on there, and this is going to come from another chemical. So if you have a phosphate donor, it's just going to plop it right onto there. Well, not just going to, it'll do it with the help of an enzyme. The other way to phosphorylate, well, there's 2 other ways. This one is oxidative phosphorylation. And in oxidative phosphorylation, electrons are transported or transferred from one electron carrier to another along an electron transport chain system. This must be done on a membrane because eventually it'll release energy to generate ATP. Chemiosmosis is the term for the process where ATP is generated from ADP using the energy derived from the electron transport chain. Now, this is just an overview. We are going to go through this in more detail so that you understand why you need a membrane to generate that energy at the end of the flow of electrons. Again, we're going to go into this in more detail. This is an overview of another way to phosphorylate, photophosphorylation. Photophosphorylation occurs only in photosynthetic cells that must have light-trapping pigments, known as chlorophylls. This light energy is converted to chemical energy in the form of ATP. During the transfer of electrons, or oxidation from chlorophyll as they pass through a system of carrier molecules. So the different ways that we can put that high energy phosphate bond onto ATP includes substrate level phosphorylation. It's coming from one substrate, one chemical to another. We can have oxidative phosphorylation, where we're going to use an electron transport chain with oxygen as the final electron acceptor. And we can have photophosphorylation, where we are using light to stimulate an electron transport chain and create the ability to put that phosphoryl group onto the ADP. So now we're going to look at these in more detail, unless I have more and all these preparatory slides I forgot about. So the metabolic pathways of energy production are a series of enzymatically catalyzed chemical reactions. They're going to extract energy from organic compounds and store it in the form of ATP. Okay. I don't know if we're into it yet. Yes, finally. Okay. Carbohydrate catabolism. So let's start. Carbohydrate catabolism is the breakdown of carbohydrates to release energy. And it's going to typically occur in 3 principal stages: glycolysis, the Krebs cycle, which I call the citric acid cycle. I don't like to call it the Krebs cycle. I will probably change that, but your book likes Krebs. And the electron transport chain. Why I like citric acid cycle? That's because it's the first product of the cycle. And I had to memorize all the steps back in the day when I took biochemistry. Krebs is just the name of the guy, so that didn't help me any. So I prefer a more descriptive name. But these 3 pathways, these 3 stages of breaking down glucose, are going to result in the extraction of a lot of energy. So we're going to start with glycolysis. Glycolysis is the oxidation of glucose to pyruvic acid, which will produce ATP and NADH. So let's look at that overview.
The metabolism of glucose is of central importance to all living organisms and is in fact performed in almost every cell. As one of the major sources of energy in living systems, glucose is made and broken down constantly in living cells. The metabolic pathway for the breakdown of glucose is called glycolysis, from the Greek glucose for sweet or sugar and lysine for breaking apart. Glycolysis is sometimes called the Embden-Meyerhof pathway after its discoverers. Glycolysis consists of 2 main phases, the preparatory stage and the energy conservation stage. The preparatory stage uses 2 molecules of ATP to phosphorylate glucose, making it easier to break it apart into 2 3-carbon molecules. In the energy conservation stage, 4 ATP and 2 NADH molecules are produced, along with 2 pyruvic acid molecules. At the end of glycolysis, the energy found in glucose has been converted to a net of 2 ATP molecules and 2 NADH molecules.
Okay. So one of the not discussed yet importance about glycolysis here is take a look at those little C's. Those are the carbons, and they're like black circles. When you start with glucose, you can count, there's 6 of them, 1, 2, 3, 4, 5, 6. And it stays as 6 through the whole preparatory phase, and then at the end is split into 2 3-carbon molecules. Those 2 3-carbon molecules, they're glyceraldehyde 3-phosphate. We'll just go with there's 2 of those. They go into the energy stage. That's where you're going to start to make energy once you have those 3. Every glucose molecule from here on out, you go times 2 because you've got 2 3-carbon molecules moving forward. So here we have the preparatory stage where 2 ATPs are used, and they're using that first step with the 6 carbon molecule to put a high energy phosphate on. Is that glucose 6-phosphate? That activates that 6 carbon molecule to move forward in the pathway. And that glucose is then split to form 2 molecules. And here, I wrote it all out. That we have glyceraldehyde 3-phosphate and 1 dihydroxyacetone phosphate. But that dihydroxyacetone phosphate is very quickly converted back to glyceraldehyde 3-phosphate. So you can just think of it as moving forward with 2 of the 3 carbon molecules and not really worry about what it specifically is. So we have those 2 glyceraldehydes. And so it's carrying those phosphates on either end. So we used energy here to make glucose 6-phosphate, put a phosphate on one end. And then we made fructose 1,6. We put a phosphate on each end, and then we split it apart. Now that we've split it apart, we're going to move down ×2. And we're going to take that phosphate off. And every time we take that phosphate off, we're donating it to an ADP to make ATP. So in that energy conserving stage, the 2 glyceraldehyde 3-phosphate molecules are oxidized to 2 pyruvic acid molecules. Remember, we're ×2, so a total of 4 ATP are produced and 2 NADHs are produced. And we end with 2 times that pyruvic acid at the end. So phosphates come off, they go on to make ATP. And at the end, we've got a nice little pyruvic acid molecule ready to move forward.
During the preparatory stage of glycolysis, energy is invested in the form of ATP to prepare glucose for utilization by the cell. This stage can be further divided into the energy investment stage, during which the ATP is used to phosphorylate glucose, and the lysis stage, during which the doubly phosphorylated glucose molecule is cleaved into 2 phosphorylated 3-carbon sugars. Let's look at the energy investment stage first. While glycolysis results in a net gain of available energy for the cell in the form of ATP and NADH, energy must first be invested to begin the process. This can only occur in a living cell with ATP available to invest. In the first step of glycolysis, a phosphate group is transferred from ATP to glucose. In the second step of glycolysis, the atoms of glucose 6-phosphate are rearranged to form fructose 6-phosphate. This rearrangement prepares the molecule for the third step of glycolysis, in which further energy is invested in the form of ATP, forming fructose 1,6-bisphosphate. In the fourth step of glycolysis, fructose 1,6-bisphosphate is cleaved into 2 3-carbon sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This step is the lysis step for which glycolysis is named. In a very rapid reaction, dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. The favored product in the reaction is actually dihydroxyacetone phosphate, and the enzyme for this step catalyzes the conversion reaction in both directions. However, since the subsequent steps of glycolysis deplete the concentration of glyceraldehyde 3-phosphate available in the cell, more dihydroxyacetone phosphate is available to be converted than glyceraldehyde 3-phosphate, and the reaction is driven toward formation of glyceraldehyde 3-phosphate. In the sixth step of glycolysis, glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglyceric acid. Remember that each glucose molecule fed into the pathway results in the production of 2 molecules of glyceraldehyde 3-phosphate, and therefore 2 molecules of each of the products from this step onward. The phosphate in this reaction does not come from ATP, but rather from a free inorganic phosphate in solution. During the oxidation of glyceraldehyde 3-phosphate, 1 NAD is reduced to NADH for each of the 2 1,3-bisphosphoglyceric acid molecules formed. During the energy-conserving stage, the 2,3-carbon sugars are oxidized over several steps to 2 molecules of pyruvic acid. Through these reactions, 2 molecules of NAD are reduced to NADH, and 4 molecules of ATP are produced. Since phosphate groups are transferred directly from the metabolic products to ADP, this ATP generation is called substrate-level phosphorylation. In contrast, in a process called oxidative phosphorylation, aerobic organisms use a proton gradient to drive the phosphorylation of ADP to ATP using free inorganic phosphate molecules. In the 7th step, the phosphate groups added in step 6 are used to phosphorylate ADP, converting 1,3-bisphosphoglyceric acid to 3-phosphoglyceric acid, and producing 1 ATP molecule for each molecule of 1,3-bisphosphoglyceric acid. The net change of the molecule from the glyceraldehyde 3-phosphate at the beginning of step 6 to the 3-phosphoglyceric acid at the end of step 7 is the oxidation of glyceraldehyde 3-phosphate. This oxidation provides the driving force to form NADH and ATP of steps 6 and 7. In step 8, the remaining phosphate groups are moved to the middle of each molecule, forming 2-phosphoglyceric acid. Each 2-phosphoglyceric acid molecule then loses a water molecule, forming phosphoenolpyruvic acid, or PEP. In the final step of glycolysis, each phosphoenolpyruvic acid molecule donates its phosphate group to an ADP molecule, forming an ATP molecule. These final 2 ATP molecules bring the net yield for glycolysis to 2 ATP molecules and 2 NADH molecules.
So there's our net at the end of glycolysis. There's glucose plus 2 ATPs, and you end up with your pyruvic acid and your 2, oh sorry, I was supposed to say times 2 at the end there. So you have pyruvic acid times 2, 4 ATPs, and 2 NADHs. And you have an overall net gain of 2 molecules of ATP for each molecule of glucose oxidized. That's because we have to use 2 at the beginning of it. I don't know if there's a table. Nope, they don't have a table, but some additional pathways that we have to glycolysis include the pentose phosphate pathway. Pentose is for 5. So in the pentose phosphate pathway, we break down 5-carbon pentose sugars or glucose, so you bring them both in together to produce NADPH. And this pathway operates simultaneously with glycolysis. And a big thing for this particular pathway is to provide intermediates for synthesis reactions. So this is an important pathway, a preparatory pathway, think of that, for a lot of anabolic reactions that we have. The enter-Deuteroff pathway is another alternative pathway. It produces NADPH and ATP. It doesn't involve glycolysis. It does operate independently. And we see it very specifically in different types, families of organisms, such as Pseudomonas, Rhizobium, and Argobacterium. These are, generally speaking, these are microbes that live in nature with plants. So they are going to occur in these metabolic pathways where they are in symbiosis with nodule plants, very often like potatoes and lima beans, soybeans. All right, on to cellular respiration. So cellular respiration is when we have the oxidation of molecules liberates electrons to operate an electron transport chain. The final electron acceptor comes from outside the cell and is inorganic. Aerobic respiration uses oxygen as the final electron acceptor. whereas anaerobic respiration uses a molecule other than oxygen as the final electron acceptor. So we'll find this in a number of microbes that are using heavy metals and such or other elements like sulfur. They'll use those as their final electron acceptor. And ATP is generated by oxidative phosphorylation. So we had our glycolysis. Glycolysis does not need oxygen. That's why it occurs in almost every living thing. Cellular respiration, on the other hand, is going to be specific towards a type of cell. And we're going to have our aerobic and anaerobics. We're going to start with the aerobic. In aerobic respiration, we have the Krebs cycle, also known, my CO2 got a little wonky off there. Krebs cycle, also known as the citric acid cycle. The pyruvic acid from glycolysis is oxidized, and we have decarboxylation, which is the loss of CO2. I'm sorry, that's over there. It's carbon dioxide occurs. So basically what that says is we're going to have a release of carbon dioxide as a byproduct. And the resulting 2-carbon compound attaches to coenzyme A, forming acetyl-CoA and NADH. So pyruvic acid has to be prepped to enter the Krebs or citric acid cycle. There we go, that's better. The oxidation of the acetyl-CoA will produce NADH, FADH2, and ATP, and again will liberate carbon dioxide as waste. So this is the entire cycle. At the very top, you see pyruvic acid is converted into acetyl-CoA. The CoA comes off, and the very first product in your citric acid cycle, in the Krebs cycle, is citric acid. That's why I like to call it citric acid. And then we move through all the different steps. You can see NADH is released. So we have that NAD being converted into NADH. And you count that. We have 1 in the preparatory stage, then in step 3, step 4, and step 8. And then we have FADH coming off in step 6. And we make some ATP down at the very bottom of our cycle there, from succinyl-CoA to succinic acid. That's not a lot of energy that's coming out of the citric acid cycle, just a couple of ATPs. Where we are going to see the production of an enormous amount of energy is in that electron transport chain. And we're going to do that in the electron transport chain really using these molecules, the NADH and FADHs. They're going to move over there and carry out some of those processes we were talking about earlier, where we're donating electrons and pulling off the hydrogens. So let's take a look at the cycle before we get into our real energy maker, the electron transport chain.
The Krebs cycle, or citric acid cycle, is one of the 3 stages of cellular respiration. During respiration, cells completely oxidize metabolites to carbon dioxide and water to obtain chemical energy. However, pyruvic acid generated by glycolysis cannot enter the Krebs cycle directly. First, it must be decarboxylated in a step that's often called the bridge or transition step. During the bridge step, 1 carbon atom is removed from pyruvic acid, forming acetate. It is then attached to coenzyme A, resulting in a complex called acetyl-CoA. Acetyl-CoA can then enter the Krebs cycle. The Krebs cycle is a carefully choreographed process that efficiently extracts the energy in the pyruvate molecule in 8 enzymatic steps. The energy is released in the form of the electron carriers NADH and FADH2, and the energy carrier guanosine triphosphate, GTP. Ultimately, during one pass through the bridge step and Krebs cycle, enough energy is extracted from a single molecule of pyruvic acid to generate 15 molecules of ATP.
Okay, so here we are. The steps.
In the first step of the citric acid cycle, acetyl-CoA donates the acetyl group to oxaloacetic acid to make citric acid. The high-energy bond between the acetyl group and coenzyme A makes the addition of the acetyl group to oxaloacetic acid possible. In the second step of the Krebs cycle, citric acid is rearranged to form isocitric acid. During this reaction, water is removed from one carbon atom of citric acid and then added back to the adjacent carbon atom. This rearrangement is necessary to prepare the molecule for 2 consecutive decarboxylation steps, both of which will generate usable energy. In the third step of the Krebs cycle, the isocitric acid is oxidized and decarboxylated to produce alpha-ketogluteric acid. The free energy released during this step is used to reduce an NAD molecule to generate an NADH molecule. The NADH molecule can move on to generate 3 ATPs through oxidative phosphorylation. In the fourth step of the Krebs cycle, alpha-ketogluteric acid loses another carbon atom in the form of carbon dioxide. Since the loss of a carbon dioxide molecule is accompanied by a large release of energy, another NADH molecule is generated from NAD, and the remaining 4-carbon succinyl group is attached to coenzyme A, producing succinyl-CoA. This coupling step allows even more energy to be extracted from the succinyl molecule. In the fifth step of the Krebs cycle, water reacts with succinyl-CoA, releasing coenzyme A and producing succinic acid. Breaking the high-energy bond between the succinyl group and CoA releases a large amount of energy, which is coupled to the phosphorylation of a guanosine diphosphate molecule to produce guanosine triphosphate. The GTP carries the same amount of energy as an ATP molecule, so it can be used to produce an ATP molecule. The coenzyme A can be recycled back to the previous step to react with alpha-ketogluteric acid. The sixth step of the Krebs cycle involves another oxidation reaction, in which succinic acid loses 2 hydrogen atoms to form fumaric acid. FAD gains these 2 hydrogen atoms, reducing FAD to FADH2. The FADH2 molecule has the reducing capability to produce 2 ATP molecules in the oxidative phosphorylation process. In the seventh step of the Krebs cycle, the newly created double bond of fumaric acid is hydrated to form malic acid. This step does not produce any energy in itself, but it prepares the intermediate for the next step, which is an oxidation step that will produce energy. In the eighth and final step of the Krebs cycle, a full cycle is reached. Malic acid is oxidized to regenerate oxaloacetic acid, one of the reactants for the first step of the Krebs cycle. At the same time, a molecule of NAD is reduced to NADH.
All right, so now we get into where we're going to make most of our energy, which is in the electron transport chain system. It must occur in a membrane, and in prokaryotes, they don't have a mitochondria, so this electron transport chain will occur in the plasma membrane of prokaryotes. Whereas in eukaryotes, this will occur in the inner mitochondrial membrane. There's a series of carrier molecules, which we can see in the picture. They've just made shorthand of these, but they're flavoproteins, so that's FMN down there, cytochromes, and ubiquinones. And these carrier molecules are oxidized and reduced as electrons are passed down the chain. At the end of the chain, that blue transmembrane protein that you see, that is an ATPase, and it's going to produce a lot of ATP through chemiosmosis. So in the next slide, we talk more about this chemiosmosis. So electrons from NADH, and I should have put in here also FADH. So FADH goes straight into that little Q. You see that guy there? So that's called complex 2. It's going into there. We reduce it, and then it goes into complex 2. Very scientific. Complex 1, complex 2, complex 3. But these electrons from the NADH and FADH will pass down the electron transport chain, And these protons, remember those H's are always attached to the electrons. So while the electrons are passed down the membrane, those H's are pulled off of the NADH and FADH and pumped through the system. And you can see them show up on the other side of the membrane. This establishes a proton gradient. So remember when we talked about all those transport processes through the membrane on Monday, and how if you have a high concentration on one end, they can flow very easily down to the other side of the membrane. It's a downhill force. That's what we're doing here. We're creating a gradient. We're creating a high concentration of hydrogens on one side of the membrane. And then when they flow back through, these protons in higher concentration on one side of the membrane are going to diffuse back through ATP synthase, and that will release energy to synthesize ATP. It's really cool to look at the actual protein structure of this ATP synthase. So if we look over on our right-hand side at that figure, you have it in blue. You see the protons coming through. They come through the channel, and then that part that says ATP synthase, it moves like a carousel. It really does. So every time you drop a hydrogen through, it does a turn. And every time it does a turn, it puts a phosphate onto ADP. So this is why it is so important that we are creating the NADH and FADH in the citric acid cycle. We don't create a lot of energy in the citric acid cycle. But by creating those NADHs and FADHs, they go to the electron transport chain, donate electrons, and pump their protons through to create that higher energy gradient and power ATP synthase. That makes a lot of energy. And that actually, the final electron acceptor in the electron transport chain is molecular oxygen, O2. Let's see how much energy we make. Each NADH can be oxidized in the electron transport chain to produce 3 molecules of ATP. And each FAD can produce 2 molecules of ATP. So do the math. We end up with a glucose plus oxygen, and you have to have your ADPs to start out. You're going to make 38 ATPs at the end of the day, and also release carbon dioxide and water. So that's a lot of energy that is made at the end of this electron transport chain. This table is table 5.3 from the new book. If you are using the old book, this table you'll be able to pull up very easily on this on the PowerPoint. This is in a PDF form when you pull up the PowerPoints. So you can go back and see exactly how much is created in each step. But I really want that take-home message to get through that NADH and FADH are creating that proton gradient in the electron transport chain to power ATPase and make a lot of energy. Okay. Anaerobic respiration. We're not going to talk a whole lot about anaerobic respiration right now. I mean, we'll move into fermentation and such, but basically, the final electron acceptor in the electron transport chain is not oxygen. It could be sulfur. It could be, gosh, I'm trying to think of other things. Copper. It could be a lot of different things. It's going to yield a lot less energy than aerobic respiration. It's very hard to culture anaerobic microbes because they don't create a lot of energy. They don't have high metabolisms. It's hard for them to grow in a way, so they're just very, very slow. And there's some examples of different electron acceptors. I guess I could have looked at the graph instead of trying to think of them. But so there's the sulfate group there. There's a nitrogen group. There's another carbon. So fermentation is one of these pathways that will release energy from the oxidation of organic molecules, but it does not require oxygen. And it's not going to use the Krebs cycle or the electron transport chain. It'll use an organic molecule as the final electron acceptor, and it produces only small amounts of ATP. Now, what fermentation really is going to do is it's going to release a lot of byproducts. Usually these byproducts are useful in industry. We're going to make cheese, you're going to make alcohol, you're going to make soy sauce. Fermentation can be used for any of these things. So here we have the different types of fermentation. We have lactic acid fermentation, where we produce lactic acid. And there's two types of this lactic acid fermentation. Homolactic fermentation, where you're just solely producing the lactic acid. And heterolactic fermentation, where we produce lactic acid and other compounds, usually ethanol. And glucose is oxidized to pyruvic acid, which is then reduced by NADH. So those are our two different pathways. This is very often how we're going to make cheese. Alcohol fermentation will produce ethanol and CO2. Glucose is oxidized to pyruvic acid, and pyruvic acid is converted to acetaldehyde and CO2. NADH will reduce the acetaldehyde to ethanol at the end. So one of the things, if you've ever seen or had, say, a beer, it's very fun to notice how there's all these little bubbles in it, right? CO2. Interestingly, that CO2 that you see in an alcoholic drink is not naturally produced. And that's because in the alcohol fermentation process, where you have yeast and it's actively going away in a big vat, it's in an anaerobic state. It's producing the CO2. You have to vent the CO2. So you actually have to have this vent and it'll bubble off. If you don't do that, it'll explode. So most of the fermentation that humans did throughout history using Saccharomyces cerevisiae, the yeast, did not have any carbonation in it. And we added that later on when we learned how to synthetically put carbon dioxide in. So this is a slide just to basically show the different kinds of fermentation that we can have, the fermentation end products based on the type of organism. So table 5.4, again, this is 5.4 in the new book. And again, you can access this through the PowerPoint. These are some different industrial uses for different types of fermentations. I'm not really going to go through this too much because we are really running out of time. We're pushing the envelope today. But you can see it. There's just a lot of really great things that we can make using fermentation. So let's briefly talk about lipid and protein catabolism, so breaking down lipids and proteins. Proteins are degraded by extracellular proteases and peptidases into amino acids. Amino acids will cross the plasma membranes. They are then deaminated, which means you take away the amine group, decarboxylated, take away the carboxyl group, desulfurized, if they have a sulfur group, only 2 of the 20 amino acids have sulfur groups, to obtain molecules that can enter the Krebs cycle for further processing. So proteins are broken down into amino acids. The amino acids are broken apart just a little bit. We take the amine, the nitrogen, which goes for waste. Carboxyl group can actually be reused. The sulfur can be reused. And then what's left of that amino acid will enter the citric acid cycle at different stages depending on the type of amino acid. Whoops. Lipids, I guess I don't have a lipid slide. That's fine. We don't need to go into too far of details, but when we break down fatty acids, you're breaking down a fatty acid, you're going to have a glycerol head, which can enter into glycolysis. And the rest of that fatty acid chain is going to go into acetyl-CoA to be prepped to enter the citric acid cycle. Take-home message, proteins and lipids, they do have other components that have to be taken off, but they are going to enter glycolysis and the citric acid cycle at different stages, just depending on their structure, and be broken down the same way as glucose. All right, so our biochemical tests and bacterial identification. Biochemical tests will identify bacteria by detecting enzymes. For example, those that are involved in that decarboxylation and dehydrogenation steps that we've talked about for amino acids. So we can use biochemical tests to figure out what kind of bacteria are in there on their metabolics. And we're going to do that. I'm actually developing some at-home labs that we can do. I think I'm going to have you guys grow some mold, and then you're going to test some of the metabolics. One thing would be to add hydrogen peroxide, a drop of hydrogen peroxide, and see if it bubbles. If it does bubble, that means you have the enzyme that breaks that down. So we'll do some of these biochemical tests, and this is taking advantage of understanding the metabolics of a microbe. You may also do the fermentation test, where we have bacteria that catabolize carbohydrate or protein to produce acid. And this can cause a pH indicator to change color. This can also be used with a Durham tube to detect gas production during fermentation. That Durham tube is that little glass tube that you see in there. If you get a bubble in there, you know that you have gas production. That's a very common test that we use for chloroform bacteria, which are chloroform bacteria are found wherever you have fecal matter, and so that's how we test drinking water to see if it's clean. The oxidase test will identify bacteria that have cytochrome C oxidase, and that's where we're trying to tease out, is this a Pseudomonas bacteria? So all of these tests are taking advantage of knowing your metabolics, and I'm trying to develop a few that you guys will be able to do on your own. Photosynthesis. Do we have time? We don't really have time to get into photosynthesis. I'm going to give it a shot. If you need to leave the lecture, feel free to. I will continue to record and try to get to the end of this lecture and then upload it for you. Photosynthesis. We have light-dependent light reactions. Oh, my CO2 got goofed around in there again. I'm sorry. Where we have the conversion of light energy into chemical energy, ATP and NADPH. There are also light-independent dark reactions where that ATP and NADPH produced from the light-dependent reactions, those are used to reduce CO2, which, sorry, it moved over a bit, carbon dioxide, to sugar. This is known as carbon fixation, and it's done via the Calvin-Benson cycle. So we're going to look at this in more detail in the following slides. Different types of plants, algae, and cyanobacteria are going to use light energy to produce sugars and oxygen. We also have anoxygenic, the very specific purple sulfur and green sulfur bacteria that are going to use light energy. They're not going to produce oxygen at the end, but they do fix carbon. So they take carbon dioxide out of the atmosphere, turn it into a sugar. They're not releasing oxygen, but they are releasing a sulfur. That's the final electron acceptor. There's a lot of different microbes that fix carbon dioxide. And we use them actually in a number of different industrial uses. I don't know if anyone's ever been over to the to Colstrip and seen some of those big coal producing factories that they have there. So they're clean burning coal. And it's really cool because some of the ways that they're capturing the CO2 is using these bacteria. They're actually grown and produced in part of the filtration system, and they're fixing the carbon. I just think it's neat to be able to use microbiology like that. All right, so let's do the quick animation.
Phototrophs are organisms that use light as their energy source and carry out photosynthesis. Photosynthetic bacteria can be divided into two groups based on their carbon source: photoautotrophs, which use carbon dioxide, and photoheterotrophs that use organic compounds. Photoheterotrophs include the green and purple non-sulfur bacteria. The photoautotrophs can be divided into two groups based upon their source of reducing power: the cyanobacteria, which use water, and the green and purple sulfur bacteria, which use sulfur compounds instead. The cyanobacteria are the prokaryotes that are most like eukaryotic algae in plants. Cyanobacteria have chlorophyll A and other pigments attached to thylakoids. This makes the cyanobacterium look very much like a eukaryotic chloroplast. During the light reaction of photosynthesis, cyanobacteria use water to reduce carbon dioxide and give off oxygen, just like eukaryotes. Therefore, both groups of organisms are said to be oxygenic. For this reason, they're usually found in an aerobic environment, such as the surface of a lake or ocean. The green and purple bacteria are so named because of the types of bacteria chlorophylls they possess. Bacterial chlorophylls absorb light at different wavelengths than chlorophyll A. In the green bacteria, the bacterial chlorophyll is located in a spherical structure called a chlorosome that lies just inside the plasma membrane. Instead of chlorosomes, the purple bacteria have their bacteria chlorophylls located on intracytoplasmic membranes, often found in layers. The green and purple bacteria do not use water as their reducing power, and thus do not produce oxygen. They're said to be anoxygenic. Therefore, the green and purple bacteria are found in anaerobic environments. The purple bacteria exist in two groups: purple sulfur bacteria and purple non-sulfur bacteria. Though they share similar pigments, the two groups differ in their carbon source and reducing power. The purple non-sulfur bacteria utilize organic compounds such as alcohols, acids, and carbohydrates for their carbon source and for reducing power. The purple sulfur bacteria, on the other hand, use carbon dioxide as their carbon source and hydrogen sulfide or thiosulfate as their reducing power. The green bacteria exist in two different groups: green sulfur bacteria and green non-sulfur bacteria. These two groups share similar pigments but differ in their carbon source and reducing power. Like their purple counterparts, the green non-sulfur bacteria use organic compounds as their carbon source and reducing power. In contrast, the green sulfur bacteria are photoautotrophs, using carbon dioxide as their carbon source and reducing it with hydrogen sulfide.
All right, so let's get into how photophosphorylation works. Now there's two photosystems, and there's photosystem 1, which is cyclic photophosphorylation. And in cyclic photophosphorylation, light excites the chlorophyll, generating high energy electrons that are passed through the electron transport chain to phosphorylate ADP to ATP. The electrons are recycled back to the chlorophyll center. Interestingly, photosystem 1 is actually the second system that works. Oh gosh, I don't know if we have time for this guy. We'll go back to that. Non-cyclic photophosphorylation is photosystem 2, and it actually occurs first. Why they're named like this is simply because of when they were discovered. Photosystem 1 was discovered first. In non-cyclic photophosphorylation, light excites electrons in the chlorophyll center of photosystem 2. Excited electrons are passed through the electron transport chain to power the phosphorylation of ADP to ATP. Electrons are passed to the chlorophyll center of photosystem 1. All right, we'll go back to this guy then. Let him talk for a minute. Oops, went too far. Okay.
Photosystem 1 is composed of a large number of chlorophyll molecules, which harvest light and pass the excited electrons between the chlorophyll molecules until the excited electrons reach the reaction center. After the excited electrons are passed to the reaction center, these electrons are passed to the next protein in the electron transport chain. In the cyclic photophosphorylation pathway, the electrons that are released by the excitation of photosystem 1 pass through 3 sets of electron transport proteins, finally returning to photosystem 1. On their way, the electrons help establish a proton gradient as they pass through a group of proteins called cytochromes. The proton gradient in turn drives ATP synthase to produce ATP through chemiosmosis.
Okay, so there's our cyclic photophosphorylation, our non-cyclic photophosphorylation, which is photosystem 2.
The non-cyclic photophosphorylation pathway utilizes both photosystem 1 and photosystem 2, and it produces the reducing molecule NADPH, as well as indirectly producing ATP. Unlike the cyclic photophosphorylation pathway, where the electron source and final destination are both photosystem 1, the electron source of non-cyclic photophosphorylation is water.
All right. So once we've created energy, we've created a lot of ATP and NADPH, now we enter the Calvin-Benson cycle, which is the light-independent phase of photosynthesis. So ATP generated from photosystem 1 and 2 are used to fix carbon. 3 carbon, CO2, carbon dioxide, is used to synthesize glyceraldehyde 3-phosphate. Now you remember now glyceraldehyde 3-phosphate, we talked about that with glycolysis. That was in the intermediate step before you start to make energy. So now glyceraldehyde 3-phosphate in the Calvin-Benson cycle may be used in gluconeogenesis to make sugars or other sugar generation. Alternatively, glyceraldehyde 3-phosphate may be converted into ribulose diphosphate using ATP as a cyclic recycler. When we look at this cycle, you see your 3-carbon molecule going around, and using energy, we can make that ribulose diphosphate, which is a 6-carbon. And that's very important. We have to continue to make some of that to keep the cycle going. When we look at our output at the very bottom, we are taking that glyceraldehyde 3-phosphate, some of our generated 3 carbons, and we're going to make sugars.
The Calvin-Benson cycle, sometimes called the light-independent reactions or dark reactions of photosynthesis, is the enzymatic pathway that uses the chemical energy of ATP and the reducing power of NADPH to generate glucose and other sugars from water and carbon dioxide. Through 2 rounds of the Calvin-Benson cycle, a single glucose molecule is produced from 6 carbon dioxide molecules and 6 water molecules, at the expense of 18 ATP molecules and 12 NADPH molecules. In the first step of the Calvin-Benson cycle, 3 carbon dioxide molecules from the air are covalently attached each to one 5-carbon ribulose bisphosphate molecule. The 3 resulting 6-carbon sugars react with water and break apart into 2 3-carbon sugars each, resulting in 6 3-phosphoglyceric acid molecules. This step is called the carbon fixation step. In the second step of the Calvin-Benson cycle, 6 molecules of ATP are used to phosphorylate each of the 3 phosphoglyceric acid molecules produced in the first step, resulting in 6 1,3-bisphosphoglyceric acid molecules. 6 NADPH molecules are then used to reduce the 1,3-bisphosphoglyceric acid molecules to 6 molecules of glyceraldehyde 3-phosphate, G3P. The ATP and NADPH required for these reactions are produced in the light-dependent reactions or through other catabolic pathways. After glyceraldehyde 3-phosphate is produced, 1 out of every 6 glyceraldehyde 3-phosphate molecules is diverted out of the cycle. When 2 glyceraldehyde 3-phosphate molecules have been diverted, one of them is converted into dihydroxyacetone phosphate. These 2 molecules react to produce fructose 1,6-bisphosphate, which can be used to produce glucose and other sugars. The reverse of these reactions occurs during glycolysis. 5 out of every 6 glyceraldehyde 3-phosphate molecules continue in the Calvin-Benson cycle to regenerate the ribulose bisphosphate required in step 1. 5 glyceraldehyde 3-phosphate molecules each contain 3 carbon atoms, for a total of 15 carbon atoms. Through a complex series of reactions, these 5 3-carbon molecules and 3 molecules of ATP are used to produce 3 5-carbon ribulose bisphosphate molecules, which can then proceed with another round of carbon fixation.
All right, so another table here, table 5.6 is available in the new book, or again, available on the PowerPoint. We have photosynthesis that's compared in different eukaryotes and prokaryotes, and just showing, so we have the eukaryotes, which are algae plants. The prokaryotes, we have a few here, cyanobacteria, green bacteria, purple bacteria. Now there's a lot of other ones out there that are not listed here that also fix carbon. I think it's actually pretty fantastic that we have so many microbes that are doing the job of pulling CO2 out of the atmosphere. And for us to harvest that, to use that, is just pretty neat. So I'm not going to go through the whole thing. We're not going to talk about all the different types of chlorophyll because this isn't that type of class. But this is a nice table to refer to. So metabolic diversity among organisms. So we have a nutritional classification of organisms where the energy source for organisms can be chemical or light. When it's chemical, you go towards the chemotrophs. When it's light, you go to the phototrophs. They can be further broken down into photoheterotrophs, photoautotrophs, chemoheterotrophs, and chemoautotrophs. We're going to look at this graph in more detail on the next slides. Phototrophs use light energy to derive ATP production. Photoautotrophs use energy obtained initially from light in the Calvin-Benson cycle to fix CO2 to sugar. There are 2 types of photoautotrophs: the oxygenic, which produce oxygen, and the anoxygenic, which do not produce oxygen. The photoheterotrophs use organic compounds as sources of carbon and are anoxygenic. So they're not going to fix CO2. On the other side of the chart, we have the chemotrophs. Chemotrophs will obtain their energy from inorganic compounds and are divided into 2 groups. The chemoautotrophs Chemoautotrophs will obtain their energy from the inorganic chemicals, and they will fix carbon. They'll use CO2 as a carbon source. Energy is used in a Calvin-Benson cycle to fix the CO2. So this is an example of microbes out there that are not using light, but are removing CO2 from the environment. Chemoheterotrophs obtain energy and carbon from organic chemicals. They can be medically and economically important. We are chemoheterotrophs. So these are the organisms that are going to be releasing CO2 into the environment. The chemoheterotrophs are the producers. Most of the other ones, not the photoheterotrophs, they don't release CO2 either, but it doesn't matter. A lot of these other ones, chemoautotrophs, photoautotrophs, are going to be fixing carbon from the environment. So here we have a chart that shows this metabolic diversity. We have our photoautotrophs fixing carbon using light, photoheterotrophs using light, but they use organic compounds. They don't fix carbon. Our chemoautotrophs have an energy source of some sort of inorganic chemical, and they will fix carbon. And then our chemoheterotrophs, which will use chemical energy, so they're going to probably eat up all the sugars that the autotrophs are making. And they're going to use a carbon source, which is just any organic compound. So again, this table is available for you on the PowerPoint. So there's different metabolic pathways of energy use. I don't have a lot of words on these next slides. I'm just going to show some different examples and just talk briefly about them before we wrap this chapter up. So we talked a lot about the breakdown of sugar, glycolysis, and we looked at all those different pathways. Well, we can also make sugar. So how do we do that? It's going to use energy. It's going to have these high phosphate bonds broken. In animals, we're going to make glucose, which will then, it's called gluconeogenesis. So when you make something, it's neogenesis. And then we chain those up together to make glycogen, and we store it in our livers. Bacteria do a very similar process. They don't have livers, but they can make glycogen as well. So they'll go through gluconeogenesis, make glucose, and they'll chain it up and store it. We can also have bacteria making peptidoglycan, and they're going to pull out of the glycolysis pathway that fructose 6-phosphate in order to make the peptidoglycan for their cell walls. Some simple lipids that are made. We can pull out of glycolysis our glyceraldehyde 3-phosphate and use it as its dihydroxyacetone phosphate derivative to make glycerol. And those glycerol heads are really important because they hold the fatty acid lipids that we can pull out of our citric acid cycle preparatory phase. And you can make chains of carbons with this head holding onto it, and this will make simple lipids, either for energy storage or to go on to make membranes, anywhere you would need some kind of lipid. Not all of the time, but often we can make amino acids. Amino acids then go on to be built into proteins, which can become enzymes to do these metabolic functions. Now, it depends on the organism. Not all organisms can make all 20 amino acids. It totally depends on the organism. Some have to obtain the amino acids from another source, and some can biosynthesize them. And it depends on how many pathways they have. If you have the pentose phosphate pathway, the citric acid cycle, and the internodeurotropic pathway, you can pretty much make all 20. So we know there's at least 3 groups, 3 big families of microbes, right, including the Pseudomonas, that can do this. But for example, animals can't. We can only make what, I don't know, half of them, and we have to get the rest from our diet. So that's why I drink my diet pop, because I'm getting my phenylalanine for the day. But if you have a metabolic disorder against phenylalanine, don't drink diet pop. But most people don't, so you're getting your phenylalanine. Anyway, take home message: from these different pathways, we can make amino acids, and there are groups of microbes that can do that. Also, very important, we have to be able to make nucleotides in order to build our DNA and RNA chains. So the biosynthesis of purine and pyrimidine nucleotides can come from a number of different pathways. We have that glucose 6-phosphate can enter the entero-deuterative pathway, so that's only going to be for certain bacteria. Other microbes and higher organisms are going to actually use the phosphoglyceric acid to make glycine. That will go down to make your purine. And they'll also use the citric acid cycle to make glutamine and aspartic acid. Those are both amino acids, which can be used to make purines and pyrimidines. So finally, the integration of metabolism. In amphibolic pathways, metabolic pathways that function in both anabolism and catabolism. This is where we have a pathway that is reversible, let's put it that way. So many of these pathways will function simultaneously with common intermediates. There's often 1 or 2 steps that are called irreversible, and an irreversible step will just have 1 arrow, and that step has to be bypassed. So for glycolysis, the very first step is actually an irreversible step. They're showing it here as reversible, but it's not. And so that has 2 steps to get around when you need to make glucose in gluconeogenesis. But pretty much all the rest of the steps are reversible. So they're amphibolic pathways. All right, this is the big picture. This is our last little animation, and then I will stop the recording and upload this. So for those of you who stuck with me, thank you, and we'll see you Monday.
Now that you've learned about different catabolic and anabolic processes of microorganisms, let's see how these fit together into a complete picture. Proteins can be broken down by protease enzymes into amino acids, which can be further broken down through the glycolysis and Krebs cycle pathways. Nucleic acids can be broken down by nuclease enzymes into nucleotides that can also enter the glycolysis pathway. Polysaccharides and other sugars can be converted into glucose and further broken down through glycolysis and the Krebs cycle. Lipase enzymes break lipids into glycerol, which enters the glycolytic pathway, and into fatty acids, which enter just prior to the Krebs cycle. Fermentation is also considered catabolism, since it converts pyruvic acid into other 2 and 3 carbon molecules, as well as carbon dioxide. Recall that anabolism is synthesis of complex molecules from simple molecules. Essentially, anabolism is the reverse of catabolism. Note that synthesis of proteins, nucleic acids, polysaccharides, and lipids is the reverse of their catabolism. The pentose phosphate pathway uses an intermediate from glycolysis to produce 5-carbon sugars called pentoses. These pentoses are then useful in synthesis of nucleotides. Photosynthesis is primarily an anabolic reaction, using light as an energy source to build complex molecules like glucose from carbon dioxide.
All right, and that is the end of lecture for today.